May I request Dr. Seema Samprani, Department Head of Biotechnology and of the Faculty of Science, to please introduce our esteemed speaker for the Nobel Prize category, Physiology and Medicine.
Good morning to one and all. It gives me immense pleasure to introduce to you Dr. Subhojit Sen, the Ramalingaswamy Fellow from the University of Mumbai, Department of Atomic Energy, Center of Excellence in Basic Sciences, to all of you today. If we start Sir's educational qualification, just now as Dr. Parvati said, he is an alma mater from our Somaya Vidya Vihar. We are so proud of you, sir. Sir did his undergrads BSc in microbiology, MSc in microbiology, PhD in molecular biology from Tata Institute of Fundamental Research, Mumbai. Presently, Sir is a Ramalingaswamy Fellow, a DBT-funded program for independent research. He has been a research fellow in the Laboratory of Tumor Biology, Johns Hopkins Medical Institute, USA. Dr. Sen has worked as a visiting fellow, Laboratory of Cell and Molecular Biology, Dr. Carl Wu National Center Institute, USA. Sir has done his postdoctoral fellow and research scholar work at TIFR, Mumbai. Sir's research interests lie in unraveling basic molecular mechanisms on how the environment shapes us as genetic changes begin either within our lifetime or over short transgenerational periods, which is a phenomenon that seems to play an important role in dictating our eventual evolutionary path. His team studies molecular mechanisms of how environmental insults modulate heritable change expression, which is a hallmark of epigenetic changes. His current focus lies in undertaking how environmental insults in the early life of an individual, be it during his fetal, infancy, adolescence, or young adult stage, that translates to either behavioral changes or even early onset of an array of genetic diseases and disorders like the diabetes, obesity, degenerative diseases, and even cancer. Sir's team currently assays for molecular markers that are the very first detectable epigenetic changes in gene expression of normal cells, which get altered to heritable misregulated states, cumulatively leading to a cancer-specific epigenome. In lieu of discovering drugs that can interfere with these processes, Sir's team is developing Chlamydomonas reinhardti as a model system to screen novel epigenetically active compounds from traditional sources. Dr. Subhojit Sen today would be highlighting about the Nobel Prize in Physiology and Medicine, which was announced on 3rd October 2022. It has been awarded for the discoveries concerning the genomes of extinct hominins and human evolution. Who else but Dr. Subhojit Sen would be the right person to talk to us about the same? Over to you, sir.
Yeah. Can you hear me? Okay, I guess you'll turn it on. Hello? Can you hear me? Okay, all right. Thanks for that lovely introduction. And thank you for inviting me here for this Nobel Prize lecture. So the story essentially is something that is almost, as was said, I did my 11th, 12th year at that time. When we used to read, we used to watch these movies of the Egyptian mummy coming alive and all of these things, which were very popular in Hollywood as well as now in Bollywood, were these kind of, that we would be able to trace these journeys of these people the ancient humans through genetic means was completely science fiction at the time, right? And I feel that from a time that when we studied, when this was science fiction at the time, and now I'm here within a span of 30 years giving a talk about our story, and the factual evidence that has come from these studies is testament to Svante Pääbo's contribution. And that is it looks like a little distorted, it seems, because of the transfer of the file, but that's okay. There might be a few distortions in the slide, but essentially can we have dim the lights here so that way we can see the, yeah. All right. So Svante Pääbo's discovery is essentially about the quotation, the citation states it was for his discoveries concerning the genomes of extinct hominins and human evolution. And so I'm going to take you through a journey of how his discoveries were brought about, and no scientific discovery rests totally on one shoulder. So I'll also try to present to you how there were several discoveries that eventually led to Svante Pääbo's getting this Nobel Prize, and how he managed to achieve what he did. Okay, so let me start by just putting up this slide here and asking you, if you can see this from a distance, asking you to identify from a show of hands. Can anybody identify if there's an Indian person in the crowd? From a show of hands, can we have that? Anybody who is able to identify an Indian person in the crowd on this picture? Okay, I see one hand, two hands. So what were you looking for? Let me ask you, what were you looking for? How did you manage to find it? Brown color, skin color? So where did you spot it? On a clock position, can you tell me? Right. So here, there is this person here that very likely is, looks like Indian. And so from a crowd, you can pick out where this Indian looking person is. That tells you something about what we are looking for, what kind of similarities, what kind of dissimilarities, even from such a large crowd, we are able to pick, right, where we are. So obviously, all of these are humans, but yet within humans, we are able to pick out which one is of a particular racial type or has a particular ethnic background, right? But if I show you this picture and tell you to pick, tell me which person is a Sri Lankan person in this image, it will be very difficult for you to do that, right? So you can see the problem here is the ability to figure out the details of the similarities versus the dissimilarities that exist in the background. It is easy for us when we are able to see this visually. And that was the essence, crux of the issue that Svante Pääbo was trying to address very early on. So this tree that I'm showing you here is kind of what I like to call the Big Bang of evolution. Okay, just like the physicists have a Big Bang, we have this Big Bang of evolution, which is a very slow Big Bang. But nevertheless, it took about, so the x-axis on this case is 4.5 billion years. Is this, yeah. If you have a laser, okay.
So maybe I can just...
No, I think the slide will change. I'm looking at the laser pointer. All right. Anyway, so so essentially this is the Big Bang. So on this axis you have the scale, right? The 4.5 billion years of evolution of the Earth, and you can see that life started very early on in the history of the Earth, and these circles that I've drawn are organisms that I have studied during my career. So I think that is why I'm able to connect, because I've been studying evolutionary wide spectrum of organisms to understand various aspects of biology. I should be able to connect why human evolution and studying all these evolutionary species, these that have evolved over so many millennia, why studying them is important. How are we all related? And what questions it can answer for us. Okay, so today I'm going to take you through that process to help you understand what Svante Pääbo achieved. So evolution, and this is part of my personal journey as well, where I try to understand the interaction of genes and the environment, right? And that is essentially the structure of biology. If you think about it, that is precisely what laid the understanding of evolution, the understanding of Darwinian evolution, gave us these two ways of understanding biology. One is that it developed so many different types of fields that helps us understand biology, and secondly, in the last 70, 80 years, it helped us understand this molecular view of understanding the same processes, which we called as the central dogma of biology. So these two, any biological problem, if you are able to address it with these two lenses, you should be able to address that problem in a wholesome sense, because there are various questions and answers that you can pose with these two aspects. So everybody has seen this image in your textbooks. All of you have seen this image in your textbooks. Can anybody tell me what is wrong with this image? So there is this chimpanzee, and then there is this ape-looking man, and then the ape-looking man has developed into another hominid type of man which is using tools, and then a Homo sapien. What is wrong with this picture? Can anybody tell me? Yeah, sure. Evolution is not linear, fair, but what is wrong with this picture? The species are not changing, and particularly, this is there in our textbooks, okay? And this is the reason why we misunderstand evolution. The chimpanzee never really turned into a human. And yet we have people who think that evolution is just a theory because chimpanzees are not turning into humans as we, in our daily lives. And so the chimpanzee never really turned into a human. The chimpanzee and human share a common ancestor, right? And that is what this journey is about. That is what Svante Pääbo's discoveries are all about. So what makes us different, and are we really more evolved? If you remember that the Big Bang that I showed you, and there were all these organisms on the surface of this Big Bang. Those organisms are all the organisms that are existing today. Everything within that bubble are organisms that went extinct or have evolved into modern organisms that we see today. So this understanding, okay, is helping us to understand how we are all related. So everything that is on this surface of this bubble are existing today and have gone through the same period of evolution, are evolved equally in terms of time. So we can't say that humans are more evolved than the bacteria. It's just that they exist in different niches, and therefore if a human can do what they can do in their niche, a bacteria can do its best job in their niche. If you put the human in the bacterial niche, the human will obviously not be able to survive, right? So this is what is the understanding of evolution, that everything on this bubble are related, and we have all equally evolved, and we are able to do our jobs best in our evolutionary niches, right? So to understand what makes us different from the rest of the species that exist today, one way to answer this is to look at our evolutionary history. So everybody knows what a family tree tells us. Everybody understands here what a family tree is. This is a typical description of a family tree where we have, in an Indian situation, we take all these names and chacha, bhatija, mama, phufa, all these names we have to describe our relationships with our families. But at the end of the day, a particular family will have a certain set of common characters which will be distinctly different from another set of families, right? So the more that you can look for commonalities in these characters, what you can gain from that information is that there is some relatedness between these people, okay? And the same thing goes for, this is another example of a family tree. This is the family tree of the Mahabharata family tree, where the Kurukshetra war and all that happened. So what it additionally tells us is the relationships, how these relationships helped, what were the crosstalks between these relationships, how these relationships emerged into new relationships. So understanding family trees is very important. Of course, I'm giving you a very current example as if these families exist today, but imagine trying to draw family trees between our dead ancients, right? Where there is no way to talk to the ancients, but you have to figure out the relatedness. You have to figure out the understandings of how things happened. So that is where Svante Pääbo's discoveries make a big difference. So it is essential to understand our relationship with our ancestors and create what are known as DNA molecular markers, because DNA molecular markers are the ones that are going to be the connecting evidence between the ancients and us today, because of that relatedness that I showed you in the Big Bang. right? So our genes are somewhere related and this based on the sequence, based on the commonality of the sequence. Just like you have common features in families, based on the commonality of the sequence, you can learn what is the oldest trait that was then passed on down and how new traits evolved, right? So genes, the sequence of DNA is what will tell us about this relatedness and how, and help us answer many questions about what we are today. So what was Paber's discovery really about was Paber started working on this feature of the Neanderthal man, right? So at the time when Paber started, there was no field of no way of studying archaic humans because DNA was very degraded in most samples. Because you can imagine, if for those of you who know about have been in a biology lab, if you go to a diagnostic lab, they'll take a couple of ml of blood to extract any kind of DNA from you to do a PCR, for example, in coronavirus or any other infection that you want to test. But now if this sample is stored for a while, and then you try to extract DNA, it becomes even more difficult. But that sometime now is about 45,000 years. just to help you understand this problem. The car mechanic asked a surgeon, I fix wirings, motors, screws and plumbing leaks all the time to keep the car well oiled and running. You do similar things on the body and yet you get paid so much more. Why so? And the surgeon answered, try doing that fixing while the car is still running, right? And so that is the problem. And so why do medical professionals are so well sorted after? They are essentially mechanics of the body, but they can do things while they have to do this while the body is running. And so it's a very difficult problem, right? So how do we understand human evolution? I put human evolution in this case, and how do we understand this? So here is an example of something like a blue thing that turns eventually into a red thing that eventually turns into blue in our body. Okay, say for example, let's say this is a red type of organism that is, say, a happy organism and eventually a moderately happy organism. But there are several steps that take place that eventually give rise to another organism, the blue organism, that is a really, really happy organism, right? So we all want to be really, really happy. So we want to know what makes us really, really happy, right? We want to be in that euphoria most of the time. So we want to understand what are the ways in which we can get there. So there are, of course, several methods, or the cell essentially uses protein machines, which we call as enzymes, which do these transition processes. But how do we understand how to go from this state to this state? We use the same problem like we did in the car. We try to pull out one of these working parts of the car and ask, does the car stop functioning or not, right? So we can take this enzyme, and if we are able to direct to that enzyme and mutate that enzyme, if we are able to create a faulty enzyme, then we can ask whether once this enzyme is mutated, where do we land? So if this enzyme is mutated, you will end up with a green person, right? So that tells you something as to, okay, if this enzyme is mutated, I'll get from here to here. So now I know at least half the path of how to get towards happiness, for example, right? But imagine now the case that you cannot do this on humans, but obviously, right? I cannot take you and start experimenting on you as to what makes you happy and what makes you sad. Let me mutate you. And that is fishing at that time, a type of methodology, a type of field where there was not much to back up himself with, right? So only 8 references in a very popular paper. And eventually they published another paper where they tried to clone this DNA, right? And after he cloned the DNA, he reported, I report here that one such clone contains 2 members of the ALU family of human repetitive DNA sequences as detected by DNA hybridization and nucleotide sequencing. So they showed that the Egyptian mummy had the same type of repetitive DNA, at least that was able to be detected. However, there's a catch. The catch is, again, by Pabo himself, and I don't think you can read that from a distance, but I'll read it out to you. Recently, Pabo has admitted now, it has become clear that a dust particle in the room is largely, which is largely skin fragments from humans. In fact, in this room, 70% of the dust particles that are roaming around in this room are shedding off from your skin. Most of the dust particles in this room is human skin. So even outside, in any crowded room or any closed room that you're sitting, most 70% of the dust particle is human skin. We are continuously shedding skin, right? So it has become clear that a dust particle in a room, largely skin fragments from humans, and a single dust particle can contain so much more DNA than the hair gram of a bone that I was trying to extract from, that gradually became clear. Essentially what he's trying to say, this is a quote from his personal story, that this DNA sequence which is published in Nature is actually a modern DNA human sequence. It was not the mummy DNA sequence at all. But it started him, so he made a mistake and he admitted the mistake later on, but this helped him start off on a journey on being able to try and better his technique so that he could get to the real DNA, right? And that is this challenge here. So what is the challenge with trying to extract DNA from fossilized remains? Now, when you all of you would have seen a dead rat or a dead lizard lying outside, and what happens to that? It obviously starts degrading, starts gathering fungus, bacteria, all kinds of things start growing on it. So as it degrades, as the tissue degrades, there is a lot of other DNA that is going to coexist with that sample. And in fact, when you extract DNA, compared to a fresh DNA, if you take 1 microgram of fresh DNA and you compare that with another ancient DNA sample, most of the DNA that you're getting is actually microbial DNA, which is designated by the gray bars. So most of your DNA are short fragments compared to the fresh DNA sample, first of all. The second problem is that most of the DNA is these gray bars, which are microbial DNA. Of this, very few pieces of DNA will be that of your actual human DNA sample, and there will be enough, even one dust particle in that sample can put enough contamination that can overwhelm your real signal. So this is the problem, right? And that is what the challenge that Pebo had to originally sort of overcome. And so, of course, they developed a lot of technologies as well as ways to handle these samples so that they could deal with contamination, and they are able to get some sequence information from such short sequences and make sense of it, right? So I'm not going to go into too many details of exactly what he was, but I'm going to now show you what kind of technological advances was Pebo's discovery sitting on, right? One of the first, which you already know, is Carey Mullis, which was a PCR, who developed PCR, is a Nobel Prize that has already been given in 1993. So Carey Mullis is an American biotechnologist, and he developed a way to amplify DNA. So that was essentially the very important step, because you can take very small amounts of DNA and in a lab now faithfully amplify that DNA. Okay, so that was the first development. The next development came from a different source, and I'm putting up his picture here. This is Bert Vogelstein. I was fortunate to be having worked in the next door lab to Bert Vogelstein while I was at Hopkins, and I know this because I was there personally. He doesn't talk about it. In fact, if you go on the internet, you will not even find this. Bert Vogelstein was one of the first one of the pioneers who developed the way you extract DNA today from plasmid kits and all of that, right? You use this silica material to be able to extract plasmid DNA using kits, right? And he has in his lab a certain box, a glass box, which is covered and kept those original samples where he had tried to first extract the DNA from, and showed the original silica columns that he had used to extract the DNA from. So he was one of the pioneers, and he was not doing it for ancient DNA. He was doing it for a very contemporary problem, which is cancer. So Bert is actually one of the first to discover the genetic basis of human tumors, okay? And also he discovered P53 as the most commonly mutated gene in most cancers. So he's actually one of the fathers. He and Weinberg are the fathers of tomorrow, if there's a Nobel Prize for this discovery, it will most likely go to him and Weinberg, right? So sample preparation was the main problem, because their lab had to deal a lot with frozen samples or fixed tissue samples, formaldehyde fixed. So the DNA was not only degraded because it's old, plus it was also chemically modified by many means. So this was a real challenge, and these were the technologies that were developed, which Pebo later was able to use to extract ancient DNA. All right, the next part that actually helped Pebo towards his discovery was the human genome sequencing project. And this was not a project by a single individual. This was actually a project by a 6-country consortium that came together, funded the sequencing of the entire human genome. So in 1953, the DNA structure was the big splash in biology, because that changed the way we look at biology. It gave us the central dogma. It gave us the understanding of how genes operate. But in 2001 is when the human genome was completed. In 2003, the first complete draft sequence was published. And within this period, we started understanding what is the blueprint that makes us who we are. What are the ABCDs of life, right? Yet, once we understand what are the ABCDs of life, once we understand the alphabets, only then can we understand what makes the words and how these words are then turned into language. What is punctuation? What is syntax? All of these will follow only once you first understand the ABCDs. And so that is what the Human Genome Project is about, right? So the Human Genome Project was, there were billions of dollars that were spent on 20 main centers which are spread all over the world in these countries. And eventually, they published what is now known commonly as the reference sequence, which was originally sequenced from 5 individuals from different ethnic backgrounds in the world. So these 5 individuals' DNA was used to generate the first reference sequence of the human genome, right? And this, this is what became the ground sort of foundation, because now once you have the reference genome, even though if Pebo was able to sequence short fragments, he would be able to hybridize them in a computer. He would be able to look at them, the identity in the computer, and figure out which part of the human genome did this come from, because he now has a reference, right? So that was the important part about the Human Genome Project. And then comes what gave the next boost to Pebo's discovery was the ability to sequence very, very small fragments of DNA as well. A part of it came from Celera Genomics, which used shotgun cloning also for the Human Genome Project. I'm not going to go into that, but particularly highlight these two persons, Shankar Balasubramaniam and David Kleneman. Essentially, they are the ones who have developed this, what you popularly know now as next-gen sequencing, or Illumina platform, right? What is also called as, technically, is called as massively parallel sequencing. Essentially, what Pebo originally had to do, remember I showed you the paper that he extracted the DNA, he had to clone those pieces and sequence them. And in that process, he ended up cloning and sequencing, in spite of the mummy's DNA, he ended up cloning and sequencing a modern human's DNA from a dust particle. So one mistake can really cost you in that procedure. But now here, the ability, what this allows you to do is to take a sample and bypass the cloning method and directly go into the sequencing method. So it's the technology of cloning is now part of the sequencing strategy itself. Okay, so that therefore allows you to sample enough number of fragments, and you don't have to worry about mistakes now. You can actually weed out the mistakes by reading the sequences. You can then figure out what are the new sequences that are coming out in your readouts. So that is the advantage of the next generation sequencing technologies that are developing, right? And so tomorrow, if there's a Nobel Prize, I hope that these two are also part of those Nobel Prizes. So, So essentially, the challenges that were solved for Pebo was amplifying small amounts of DNA because of Kary-Mullis's PCR, dealing with contamination, which his lab was able to do because they were dealing with archaeological samples, the massively parallel sequencing, and then to identify where they came from, which involved a lot of statistical information to be able to get enough information so that you can get statistically valid data, right? So I'm not going to go into the details of the slide, but if anybody is interested, I can share these slides with you. This is a slide from Professor Partha Majumdar, who's at the National Institute of Biomedical Sciences. Essentially, what kind of techniques were used for developing each of these problems is given here. So most of it, as you can see, is PCR based, and some of it is cloning based, and some of it is dealing with how you sequence modified DNA, the ancient DNA that has gotten modified over a long period of time, essentially. And then, of course, there are other problems with the types of amino acid representations and type of base representations that are there in these samples. So by the time Pebo had moved on to looking at Neanderthal samples, several archaeological samples are now being used in the modern days to sequence human DNA. They are not just bones, but also all kinds of artifacts that are part of the civilizations that grew along with these, that are isolated from the same layers that these bones are found in. Right? So these are all good samples now because of the ability to bypass these problems that were there. So what did they find out? Okay? So here is the way of how they are addressing the problem. There are two ways in which you can address how we are all related. One is what we call as maternal contribution. So the maternal contribution, which is on the left hand side, the pink bar, essentially is sequencing the mitochondrial DNA. Why mitochondrial DNA? Because mitochondrial DNA is the one that is being passed down from mother to daughter, daughter to their daughter, and so on, even sons as well. So we all get our mitochondrial DNA only from our mother and never from our father. Okay, because that's the sperm and the egg fusion, and the sperm mitochondria does not get into the egg. Only the sperm nucleus gets into the egg, and the mitochondrial contribution always comes from the egg. So that's why if you sequence mitochondrial DNA, you'll be able to trace the maternal lineage of your ancestry. Right? So that is what was studied. So essentially that defined as to when was the first Eve born. Right? If you want to put it that way, Adam and Eve, the question of Adam and Eve. And the first ancestor, which is the female ancestor, seems to be about 200,000 years old. Right? So that's probably when we define as when Homo sapiens as a species was born. And if you look at the Y chromosome, which is part of your autosomal contributions as well, so if you look at the autosomes and the Y chromosome, as you all know, is the paternal contribution, that defined the common ancestor as about 400,000 years old. So Adam and Eve were about 200,000 years apart, right? So obviously the concept that we all came from the single parent is proven false. We are a culmination of multiple such events over 200,000 years, right? So what kind of information did we get from this? So here is that family tree that I told you about, right? There are two family trees here. This is the maternal family tree, and that is the paternal family tree. But what is important for you to look at is the color coding of these trees. And these colors are part of the ancient lineage. These colors are part of the later lineage, right? And if you look at the color, the representation of those colors in these pie charts, you will see that Africa is more colored with the ancient lineages compared to, say, for example, the Americas, which is the New World. Americas is mostly homogeneous with the later markers, right? These are molecular DNA sequences that help us identify which are the common markers which are spread all over the world, and which are the new markers, new molecular sequences that have evolved of late. And by looking at these sequences, you can answer as to where we were originally from and where we spread and where we landed eventually, where we ended up landing, right? So you can say from this image itself that there must have been the early evolution in Africa, and eventually from this the diversity expanded into the other regions of new molecular markers that were developed that were much later in evolution as far as the family trees are concerned. And this is true both for maternal as well as paternal markers. All right? So this tells us that the highest, another sort of deduction from these studies by Pebo and several others was that the highest diversity, genetic diversity, because that was where it originated, was in Africa, because that's where there was a maximum diversity. Then one of these branches came out and then populated the rest of the world, and that led to, and basically if you can see more commonalities in that set of humans that came out and populated the rest of the world. So genetic diversity is actually more in Africa than the rest of the world, right? So here is now a tree just to show you that out of Africa kind of evolution. About 2 million years ago, as I said, Eve was born. About 2 million years ago is when the Homo species was born, right? Homo as a genus was born. And so there was Homo erectus, and that Homo erectus eventually gave rise to Homo sapiens and a couple of other species. Here there's Neanderthals, here there is the Bodos, and there are several humans, and this is the sapiens branch that eventually you can see that these other branches ended up, right? They ended here and they ended here. But the sapiens branch pretty much started branching into, from Africa it started branching into everywhere, Europe, Eastern Asia, as well as Australia. The Neanderthals, on the other hand, which branched away from the Homo sapiens very early on, about more than 800,000 years ago, that these Neanderthals, pre-Neanderthals, then populated the northern Europe and the branch from Europe to all the way to Eastern Europe, so Eastern Asia. So this is the Neanderthal branch, right? So the question then was, so this all came from sequence information. So the question then was that what were we, so clearly we were not alone, but then why did they all disappear? Why is it that we are only there today, and are we carrying any of their genes or not, right? So this is the first major discovery which really changed the way we think about human evolution, and this again came from Pebo's lab. Pebo, from this Vindhya cave in Croatia, Pebo was able to sequence very high quality Neanderthal DNA sequences from 3 different individuals, and those are the 3 different bones that are shown there. The 3 different individuals, and these 3 fragments were all around 40,000 years old, right? And DNA extracted from these bones was used to generate the first draft sequence of the Neanderthal genome, which was, now we know, is about 50% of the Neanderthal genome. So you already by then, this was around in 2008 or 10, I don't remember, but this was around the time when the complete draft of the first draft of the Neanderthal genome was published, around 2010, and by 2014 another lab eventually published the full sequence of the Neanderthal genome with more high quality sequences. So yes, so this is 2010, the first draft of the Neanderthal genome. So here are the few things that we understood from Pebo's discovery. First of all, Neanderthals are closer to non-Africans than Africans, right? Which means that because Neanderthals had moved out to Europe before Homo sapiens moved out of Africa, they already populated 800,000 years ago, they populated northern Europe. Right, and so when the Homo sapiens moved out of Africa, they started interacting with Neanderthals and they started mating with the Neanderthal hominids as well. And that created a exchange of genes between Homo sapiens, the early humans, archaic humans, and the Neanderthal man. So Neanderthals are closer to Africans than non-Africans, and this is how we know. Basically, Pebo defined certain markers that were unique to Neanderthal sequences, that are the red boxes or the red triangles, and he compared that to the reference sequence of the modern humans as well as archaic humans, and was able to figure out what are these markers that are uniquely Neanderthal. Right? So by looking at these reference sequences, he was able to identify what are unique. So the second thing that was interesting, the second thing that was interesting that came was that the direction of gene flow. Obviously, if a male and a female mate, they are exchanging their genes, right? But what Pebo found out from his sequence studies was that we harbor Neanderthal DNA, but if you look at any of the Neanderthal genomes, they don't harbor human sequences. We of course have a lot of common sequences, but those sequences that are unique to Homo sapiens are never found in the Neanderthal sequences. The other way around is not true. But we are harboring Neanderthal markers as well, right? So about anywhere between 1 to 2% to sometimes even 4% of our DNA of all people who are existing today out of Africa contain Neanderthal markers as well, right? So that is what came out of this study. The last thing is that the segments of Neanderthal ancestry in present-day humans can be identified with high confidence because he was able to define these markers. So by now there are several such archaic sequences that are already sequenced. So this was way back in 2010, today in 2022, close to about 8,000 archaic humans have been sequenced by now, right? Archaic hominids have been sequenced, and that is represented here in this graph as to where they come from. What you will clearly notice is that there is a blank here, right? So this stretch particularly is blank, possibly, there could be several reasons for this. What Pebo and his colleagues offer is that maybe these are tropical regions and therefore DNA is very easily degraded, right? There's another reason could be that maybe because the way we cremate our ancients, maybe because we burn them or we cremate them or we cremated them under conditions which were not very highly preservative. So therefore, we have lost that information in our ancient DNA samples, right? So it is very hard yet for us to represent this belt there's very little sampling from this belt of, here of course it's the Saharas, so deserts of course, and then here in India we find very little ancient DNA information. So this is the next discovery that put Pebo on the map again, and this was subsequently from another cave in Siberia which is called the Denisova cave, and they had found this little bone. You can see the finger, and that bone is right there on that finger. So that little bone was discovered in this cave at the time, which the discoverers of this bone, the archaeologists, were very sort of smart in identifying that this is actually a human bone. So they went ahead, they went to the Pebo started collaborating with these archaeologists, and they sequenced this little bone, and by sequencing that little bone, they discovered a very interesting discovery, that this bone is from an archaic human that does not exist in any other sample. So it was a new human species they had identified just by looking at its DNA, right? So they figured out that this was different from the Neanderthals as well as different from the modern humans as well as archaic humans. So they called it, this group started being identified as the Denisovan humans, right? And so the Denisovan species. What is interesting is that 25% of the haplotypes that were identified, so till today there are about more than 1,000 Neanderthal genomes, 8,000 total archaic humans, and very few, maybe I can count on one hand how many Denisovan genomes have been sequenced. So till today, very few Denisovan genomes have been sequenced because we have not yet found a complete skeleton of the Denisovan man or Denisovan human. So only small parts, a jawbone or a tooth or a finger bone or something like that has been found, which then by sequencing tells us that it belongs to the Denisovans and not the Neanderthal or the Homo sapiens. And that gives us certain identifying markers that tells us that if you look at the modern human genome of these mixtures that happened, the modern human genome still has certain parts that don't identify either with the archaic humans or with the Neanderthals or with the Denisovans. So what does this mean? This only could mean, and this is what Paber's another hypothesis is, that there must be another ancient human that we have, or hominid that we have crossed with, and that is remaining in our DNA and that is yet to be discovered. So that is another discovery that came from these papers, is that there is yet, our DNA sequences tell us that there is yet another type of hominid that is to be discovered, that is waiting to be discovered. And by now there are about 21 hominid species that are already human species, Homo species that are already there. So, and the last one was discovered as recently as 2018. So you are living in the times of discovery of these major happenings, right? So tomorrow, if you join one of these labs, you can be part of these discoveries as well. So if you look at the hominids, these Denisovan hominids, you'll see that they exist, these markers exist, very little of the markers exist in these populations, but they are more often there in the oceanic populations, okay, oceania populations. All right. Now, this is a 2018 paper which was published, and this is another interesting study because we keep talking about Neanderthal, as man exited out of Africa, moved out of Africa, they mated with the Neanderthals, and then we saw that there are Denisovan markers also there in the human genome. So it seemed that we were pretty much mixing with these populations, right? So what is the real evidence that we actually mixed? And that is from this paper. This paper said that there was an offspring, this bone that they sequenced, that bone that is shown there, was sequenced, and what they showed with this bone, of course I'm sure you're not able to read this sequence from far, but essentially what they did was they were trying to align which of the sequences match. The blue is the Neanderthal sequence, the orange is the yellow is the Denisovan sequence, and what matches at both is the purple sequence. So you can see from this bone, practically throughout the chromosome, you have a 50-50% contribution from each of the Neanderthal person as well as the Denisovan genome, which tells you that this individual or this bone actually came from a Neanderthal parent and from one parent being Neanderthal and the other parent being Denisovan. So this is a very interesting case where almost 50% of the DNA came from either parent, right? This was like a nail in the coffin to say that these two populations did mate, and this is the first child of that particular mating event, right? So of course that is, so what was interesting, another thing was that the mother was Neanderthal in this case, the father was Denisovan, and yet the markers that were Neanderthal are found in the Denisovan genomes and the human genomes, but never the vice versa. The gene flow, for a strange reason, was always this way, always towards modern humans or the Denisovan humans, these markers, which kind of gave us the idea that maybe the children who were born continue to live with the Denisovan communities and the modern human communities, but never back with the Neanderthal communities. That was a strange finding, but that is the only way to explain it right now. So moving on. So this is the final story of us, right? This is I'm coming to the last part of my slides. For those of you who are interested, there's this link up here which you can put on your phones, Human Odyssey Map. If you type this on Google, it will take you to a California website where you can in real time look, basically there's a slider, and by sliding across the time in temporal manner, you can see how the climate changed and how that affected the paths of migration. So there were several ice ages and shrinking of ice and growing of the ice ages across Europe and Siberia that basically dictated the way humans were passing on from one region to another. In fact, the humans moved to the New World very long ago, about 15,000 years ago, because at that time the ice age persisted and the land bridge between the Americas and Russia did exist at that time, right? So that is how you can go through this website and that will tell you how this evolution happened. So in short, to put everything down on this one slide, we all were born here. It seems to be the cradle of evolution, and we still don't know why this was the cradle of evolution, why Africa was the heaven on earth, right? We still don't know. And from there, the modern the archaic humans stepped out of Africa, and that is when they mixed with the Neanderthals, who had already moved out of Africa 800,000 years ago. Around 70 to 50,000 years ago, we moved out of Africa, and then around 45,000 years ago, or 35,000 years ago, we mixed with the Denisovans as well, and that population then migrated down towards different parts of the Oceania region as well. A separate arm had moved down towards the Australias, the aboriginal populations, way back around, again, 50 to 60,000 years ago. So this is the story of us, how we got this data. Now, question is, how are we using this data? Yes, this is a fancy story. We now know where we came from and where we originated, but how is this information all useful to us? What can we do today with this information? How can this help our careers? How can this help our medical well-being? So here is a couple of things here. So this is one of the markers that exists on a gene, which is a receptor binding protein for the prostaglandin. Okay, so here, progesterone, not prostaglandin, progesterone. So this receptor essentially lines your uterus, and the progesterone, which is released by the ovaries, is then binds to the receptor and helps the implantation of the baby. So it allows fertility to proceed, right? So once the implantation happens, then the person can get pregnant. So this receptor is the orange. There are two forms of this receptor. The blue form is the modern human form, and the orange form is the orange variant, as I would like to call it, is the Neanderthal variant. Right? And what you can see directly, even if I had not told you the story about out of Africa, just from the appearance of this variants in modern humans, you can see that it completely is absent in Africa, right? But it is present in some form or the other in the rest of the world, right? So this itself again tells you that this marker did not exist in this population, but exists now in these other populations. So again, the same ancient versus modern marker, and that tells you that, so how is this relevant to us now? So this receptor variant was associated with preterm births. So wherever this variant existed, there were premature births. It seemed like a bad thing after all, right, to have premature births. So it seems like this Neanderthal variant which is a risk to premature births, could have been one of the causes of the demise of the Neanderthal man. We don't know, right? It could be that because they carried this gene, their populations were eventually wiped out. We don't know that. But the other thing is that if it was that bad and it eliminated them, then why not these populations that are carrying it? Why is it still spreading around in the Homo sapiens? Why is it that we are still carrying this gene after 200,000 years, right? So it means that it spread rapidly. So when Pebo's group tracked this marker, they saw that this rapidly dispersed right around 6,005 years ago, which is around in modern times of civilization, right? 10,000 years ago is when we stopped being hunter-gatherers, we started settling down into as farmers. So around 6,500 years ago, this markers really started spreading across in humans, which means that it would give an advantage to that population somehow. So how is that preterm births is giving an advantage to us humans today? Right? So when Pebo's group looked at the sequence information available from the UK Biobank, now this is a public repository where all the genomes of sequences of people from UK are being deposited, so you can study those sequences and try to understand what kind of susceptibilities to disease they have or not, right? And here are these variants, for example. This variant is, but I know you can't read it, but essentially this variant is going to give you a susceptibility to hemorrhage, which is bleeding, and susceptibility to miscarriage, which means preterm births is due to miscarriage, right? So this variant is giving you these disadvantages. But it is also negatively associated with number of sisters. So what does this mean? That the Neanderthal populations suffered these problems probably. But in humans now, because we have the human variant, these human variants are probably positively associated for this phenotype, right? So that means the negative, that this variant is negatively associated with hemorrhage, miscarriages, and allows for more survival of the progeny. So you have more babies, although you might have premature babies. In the long picture of evolution, it is probably better to have premature babies and have them surviving rather than having them born later and being dead, like that, right? So so this is again all hypothesis. This is all inference that we draw from the data. This is what Pebo's inferences are from this data, right? Moving on. So this is where I'm showing you that the same gene can have both advantages as well as disadvantages, and how evolution really plays with both gains as well as losses to be able to figure out whether that gene really has to spread in evolution or not. And this is a live example of that today. Another relevant example is that of COVID-19. So when this was also published just 2 years ago, and this was during the COVID pandemic when they started looking at the data as to what makes us susceptible to the COVID-19 infection. And this is on the left hand side, this black and white plot is popularly in genetics is called as the Manhattan plot. Essentially what it is doing is that it is plotting the types of mutations that you find across all 46 chromosomes, right? The number of frequencies of these mutations is plotted here. The number of individuals carrying that mutation is plotted here. So you can see that there are some really tall skyscrapers in this Manhattan plot, right? So this means that this mutation is strongly associated with the susceptibility to COVID-19. And this mutation turned out to be a Neanderthal variant. So is it true, therefore, that maybe Neanderthals were more susceptible to corona-like diseases and therefore they were wiped out? We don't know. Right? So this is another open question. Did an epidemic like corona wipe out the Neanderthal man or Denisovan man or something like that? Right? Did that happen? So these are things that are yet to be figured out. In fact, what this graph shows is that if you have the quote unquote the modern human variant, your susceptibility to corona is around 7%. This is actually fatality due to corona. Means your chances of dying due to corona is around 7%, but your fatality increases to 13% if you're carrying the Neanderthal variant, almost double. So it is really bad for you, right? So this is the distribution of that variant, and I wanted to show you this slide particularly because we have selfish representation here, the Indian representation here, right? So now you can see that in the modern humans, this variant is largely present in India. And if you look at the representation of these haplotypes in the Indian population, Europe has about 16% carriers, but South Asia has about 50% carriers of this particular variant. So why is it so widespread in India, or rather the South Asian population? In the rest of the world, in fact, although these populations still carry the Neanderthal gene, they have lost this variant. Which means that, so what Pebo's group explains this as is maybe these groups were exposed to these coronaviruses over a long period of evolutionary time, and therefore those people who were susceptible to those coronaviruses died out in these populations and this variant was not passed on. But in here, either we were not exposed to the same coronaviruses and therefore this gene was just carried on, or this gene actually gave us an advantage. And so this is of course, I'm calling it a gene, but we don't know which gene, because this is just a random SNP or a random single nucleotide polymorphism on that particular chromosome. So what was later found out in Pebo's papers as well was that this gene is this mutation is sitting very close to another gene, interesting gene called CCR5. And what does CCR5 do? It gives you resistance to HIV. Right? So here is another thing that how we still are yet to understand this picture, but it could be that maybe this variant also has a positive side on the human story as well, not just the negative side. So this is how you have to understand evolution in terms of both positives and negatives. How do you weigh out mutations? All mutations are not just bad or ugly. Some are also beneficial. The same mutation can be beneficial to us depending on the niche that you are surviving in, right? So this variant is proposed to have caused about 1.1 million more deaths due to this variant if we rather didn't have this variant. So you can see the impact of this study in modern humans. If we knew about this variant and know how to treat it, we could have saved 1.1 million lives during corona. Right? So this kind of studies are also being done in India, by the way, and this is a study from Partha Majumdar's lab in near Kolkata in Kalyani, and there they are looking at the Andaman Nicobar, because those are very indigenous populations of the islands, and they have never really mixed with other humans for a very long time. They have been isolated for a very long time. So looking at these markers helps us understand as to when did we come to these areas and how did we, or what we are, right? So what they see in the modern human of these two, the Jarawa and the Onge tribes, which are the Andaman Nicobar tribes, they are very different looking tribes to each other, but they both have like African negroid features, right? The curly, dark curly hair, more dark skin, and all of those kind of features, and the negroid nose and those kind of features. So the green is the humans, the modern human sequence. They have some bits of the Neanderthal genome. They do have some bits of the Denisovan genome as well, but they also have these yellow pieces which seem to be another species altogether. So this seems that there's yet another, so there can be more discoveries happening within our own populations by studying these new variants, right? So there seems to be another population yet to be discovered even by these studies. So with that, what did we do better? Essentially, if you look at all the changes, if you just tabulate all the changes, there are about 30,000 single nucleotide changes in the Neanderthal versus human genome. And that's not really a lot of changes, because the human genome is a 10 to the power 9 bases, a billion bases. And in that, it's only 30,000 changes. And these 30,000 changes, all of them are not in genes that are going to be important. 100 are just small insertions and deletions all over the genome in random places. 3,000, only 3,000 are in regulatory regions, and only 100 amino acids are getting affected by the Neanderthal changes, right? So it's a very small fraction of change. If you think about it, how these changes are affecting the real proteins that do the function, those enzyme C that I talked about, right? So it is important. So basically, the tools and technologies. So how are Neanderthals therefore different from us, right? That is what this slide summarizes. Essentially, Neanderthals, if you look at them over 400,000 years of their existence, 450,000, depending on whom you talk to, over 400 years of their existence, you look at the tools 400,000 years ago and 400,000 years later, the stone tools look pretty much the same, identical across 400,000 years. But if you look at humans, within 150,000 years, our tools have evolved drastically. We have changed tools drastically. The next thing is art. If you look at, you have heard of stone, of caves art, right? Ancient art. Even in Bhimbetka near Bhopal, you have ancient human art as well in the caves. And these arts are very, there are horses and humans and cows and all kinds of animals. It's very real art. You can identify with that art. But if you look at the Neanderthal caves, the kind of art which we don't know whether Neanderthal man did it or not, but we definitely do know that these come from the Neanderthal caves, and that art is very abstract. Their art is not figurative art like cows and elephants and goats and sheep. Their arts are like lines and drawings and right angles, and they are very different art. It's abstract art. So their art was also very different. So there may be that development of cognition of how to represent their world was very different. The neural development and the neural cognition was very different from the human type. And then, of course, as I said, although Neanderthals existed for 450,000 years, they were still always in Europe. They never really migrated out of there. But look at what we did in the 150,000 years that we stepped out of Africa. In 70,000 years, we stepped out of Africa. We stepped out of Africa and we populated the entire world in 70,000 years. Right? So that is another thing that the human genes enabled us to do. And then, of course, we rapidly multiply. There's no question about that. And of course, we crossed even open waters within 40,000 years. None of the other species was really crossing open waters. It was only the Homo sapiens that crossed open waters as a species. All the other hominid species stayed on landlocked regions wherever they were. They never crossed water. So that's another interesting difference between us and the ancient hominids. So that is the story of us. And can anybody tell me what this is? What picture this depicts? Pride, right? So this is pride, and why am I showing this picture? This picture seems to be relevant to this topic, but actually it is very relevant to the two people that got Nobel Prizes this year, and that is essentially, essentially two people, Bertocci, Carolyn Bertocci, and Svante Pääbo. Svante Pääbo, in his book, The Neanderthal Man, which was published in the last decade, has a confession of him being bisexual. Bertocci is a lesbian single mother, and these are two LGBTQI plus people who are given Nobel Prizes today. For the first time, the Nobel Prize has been given to somebody from these communities as well. Right? So this is an exciting story for us. It's an exciting example that everybody who's given, if given the right fertilizer, given the right way to grow, we can excel in what we do. It doesn't matter what background we come from. Thank you.
Questions from the crowd? Any questions?
Sure, go ahead. There's a mic, if she can get a mic.
That was a really nice, vast coverage of all topics, and with quite great deal of simplicity, which we actually need. My question is, we are tracing the complexity of our ancestral genome with what exists today. So if we are tracing the footsteps, are we extrapolating what might happen in future? Are we going to have, or is there any kind of conversation going on in terms of what may happen to Homo sapiens? Is there any species that's already coming up or might come up in future, and then what's the timeline?
So we are Homo sapiens sapiens, and that reason is because we are so homogeneous in our DNA. We consist of about 10 raised to 9 base, 3 into 10 power 9 base pairs, a billion base pairs, 3 billion bases. And when our cells divide, each time they make one mistake, about, statistically speaking, 1 to 10, depending on the cell type, but at least one mistake, right? So, and if you and we are all growing from this one single cell, right? The egg, the fertilized egg, and then we are now an individual with so many more cells. So the number of divisions is about 2 to the power 48, 46 to 48, right? 2 to the power 48. Now, if you put that number, if you just compute that number in a calculator, you will see that that amounts to about 10 to the power 15 mutations. 10 to the power 15 mutations in a genome that is 10 to the power 9 base pairs. So we are accumulating mutations. There's no doubt about that. And we are constantly, evolution is not going to stop. We are not the end of evolution. We are not the pinnacle of evolution. But are we going to become a different species or not is something that, as we have learned from evolution, only natural selection will dictate. And so what we are learning from these studies is how did they disappear and why did they disappear? If we are able to figure that out, it might tell us as to what should we also be careful of. What kind of natural disasters can we also be careful of, and what can wipe us out as well, right? So why is global warming being talked about? Why is epidemics, these kind of frequent zoonotic diseases that are jumping over from other species being talked about today? Because those are the, as we can see from the talk, those could be real diseases that could wipe out very large chunks of the population. And that is precisely how natural selection works. Once you have a wiping out of the population, then you have a new species repopulating the environment in which that is left over, right? So that is my short answer to your question, that we will evolve into something else someday.
If I may have a continuation of the same, in Indian scriptures there is a reference that there is an evolution of human beings. So there is a distinct change of characteristics. If you say the human beings, now I'm not very sure exactly where, but the earlier human beings were larger in size, more apt in certain abilities, and then compared to, in terms of yuga, if I may, then the currently existing human beings are comparatively just one particular feature size-wise is much smaller. So in that situation, if we extrapolate, which particular character that may change completely or may stand out completely? Any
So first
information
of
on
all, if you look at the data of all, even if you don't worry about DNA, even if you look at archaeological data, we have grown in size. We have actually not become smaller.
Okay.
What has grown in size up till 2 million years ago was particularly our brain size. The brain to body mass ratio has been increasing. Of course, elephants and whales and the blue whale has the largest brain just because it's a very large body. But doesn't mean that larger the brain size, the more intelligent you are. It is the body mass to brain size that really matters, right? And so humans did keep increasing compared to all archaic humans. The volume of the brain did keep increasing, but until about 2 million years ago, and then it started shrinking by a small percentage. So modern man actually has only just a little smaller cranium, which may be actually not even important. Maybe it reached a particular plateau and then realized it's not really necessary to maintain that volume, so it shrank, and we can still do the things better, right? So that could be the answer. That is what I can relate to. But if you look at scriptures and you see this, I mean, I know I've heard the story that scriptures say that we have evolved from fishes to turtles. You look at the Dashavatar, it is essentially a representation of evolution, and that's not true because that leaves out many other species that were in between. So it is what were around us when we were writing scriptures. What were around us is what we picked up as visible organisms, and we developed our own scriptures, right? So of course the simpler organisms are further down in the description, and the complex organisms are on the top. But that's what evolution has taught us, that complexity need not mean hierarchy. Complexity, all these organisms, as I earlier said, are equally evolved. Humans are not better evolved than the bacteria, or humans are not better evolved than the turtle. If you put a human and a cockroach, you take a human and a cockroach and you put the human and tell them to live in a gutter, he will die out soon. He or she will die out soon. You take that cockroach and put that cockroach on the moon, will not survive. But humans can, right? We can create environments in which we can survive, but we cannot survive in the gutter. At the same time, the cockroach can do much better in the gutter than it can do on the moon. So we have to worry about these things. We have to look at these things in their evolutionary niches, not as a broad capacity of a property that has made us better than everything else.
Thank you.
Yeah. That's a very anthropomorphic way of looking at ourselves, that we are far better than everybody else. You have a question, anybody?
Hello, sir. Good afternoon. Good morning. I have two things to, rather I will say, not a doubt, but a confusion that I would like to be clear of. Like, once in one of the previous slides, you mentioned that the tree started from Africa, of course, and then it spread throughout the world. And then there was this whole idea of Denisovans, bone that was received from Siberia region. So I was confused that whether the tree started from Africa and Denisovans are the individual species, which, not species I would say right now, but were individual population that grew in the Siberia region and then they mated, or what? Like, what was the flow?
So yeah, just like Neanderthals, which moved out of Africa 8,000 years ago, 800,000 years ago, Denisovans also moved out of Africa a very long ago. We don't know exactly, I mean, I think the timelines are there in the papers, I don't remember exactly when. But those two species moved out of Africa much before Homo sapiens were moved out of Africa, or were born and moved out of Africa. These were the times when Homo erectus gave birth to Homo erectus became Neanderthals, became Denisovans. So those branches came around the same time, right, around 800,000 years ago. It so happened that Denisovans moved out and happened to settle in East Asia, and that was the region they stayed in. All the caves there have these bones. But as I showed you an example, even there was a case of a cave where there was a bone where both Neanderthal and Denisovan man coexisted, because they gave birth to a baby as well, which had 50% DNA. So yes, so these populations moved out long before Homo sapiens moved out of Africa. And then Homo sapiens, the current, what we call as archaic humans, is the Homo sapiens. They moved out of Africa and started populating, and eventually the other species died out. We still don't know why. But Homo sapiens was able to repopulate, sort of capture all the different environments of the earth, right? Populate every environment of the earth. That is why we call ourselves Homo sapiens sapien, which is modern humans. So these are the differences in terminologies. Now I just wanted to kind of correct you on one last thing, is that I mentioned, and I didn't clarify that, I did mention that the classical definition of species is the ability to form fertile offspring. But that is, from this itself we can see that cross-species habitations do occur. We also know of the liger, we also know of the donkey, the mule, and we know of several cross-species habitations that can happen both in animals as well as plants. Yes, all of them are not fertile, but if it were to give a fertile offspring, then it would lead to new speciation as well. So, the definition of species is changing in today's molecular era, and because of these studies, right? We have to give a molecular definition of species in terms of what is the similarities and differences in DNA. What is that threshold, or what is that cutoff when we call two different sequences a different species? So that is a matter of study.
Okay. The other thing I wanted to ask you is that you also started with one of the things, that the textbook photo that we have. So that's I will also agree on the point that the conveying of what evolution is like is very vague in the present curriculum. So how would, if someone from the audience or someone of us become a science educator in future sense, and how we can convey this thing in a better sense? Like, you have any idea on
That's
that?
why I show you that bubble, because everything that exists on the bubble existing today, chimpanzee and humans coexist today in the same environment. So we could not have evolved from each other because we see the family tree. It's just like saying that you and your cousin, even if they look identical, you cannot have evolved from your cousin, or you couldn't have been born from your cousin. You both have had a common ancestor, some great-grandfather who was common between both of you. So it's to identify that great-grandfather, which they keep calling the missing link. It's no longer a missing link. We see several steps of evolution that has led us today to what we are today. Right? There will be several more sub-steps in between, but we have already discovered many of these steps that led from the original primate ancestor to chimpanzees, bonobos, as well as several Homo species, which are 21 human species now.
Thank you so much.
Thank you.
Thank you for the questions. I think Dr. Subhojit said he created a blend of examples, blend of research articles, articulated a lot of research and interpretation to make you or give you insights about what the entire Nobel Prize award was all about and why it is very impactful. Apart from all the technical details, there are certain things which he meant, which was very subtle, but that should be your take-home message. For example, mistakes will cost you, but the mindset to accept it and overcome will lead you to greatness. Research is a collective effort. Cross-cultural, interdisciplinary, global networking is impactful, leads to an impactful research. He gave you a lot of examples. It also undermines a lot of statistics and mathematics involved. So you science students think that science is science, there is no inbreeding of or integration of mathematics or data analytics is nothing. It's crucial. Research is learning the art of interpreting, the way he interpreted. I mean, a lot of times there were a lot of goosebumps, like I'm a molecular biologist. For me, when he says a lot of, with examples, I think there is a spark that I can see in you as well. So definition, that's a very, very strong statement from his side. Definition of species is changing in this molecular era, and there are much that you can do. May I request, on behalf of the entire Somaya community, may I request Dr. Mayuri Argeri, the Dean of Research and Director CIRAC, to please felicitate our speaker with the moment of Jagriti. Dr. Sen, this is a memento from a part of our outreach activity from our Nareshwadi Learning Center, and they are crafted and designed by our Wali community, again, a indigenous tribe in Dhanu.
Thank you. I really like it.
Thank you, sir.
Thank you.
May I request Dr. Jacinda George, Assistant Professor, Department of Biotechnology, to please deliver the vote of thanks.
Thank you, Dr. Parvati. In the words of Sir John F. Kennedy, we must find time to stop and thank the people who make a difference in our lives. These words truly befit this moment, where I am quite certain we are all awestruck by the sensational talk by Dr. Subhojit Sen. Therefore, I commence this vote of thanks by extending a heartfelt gratitude to Dr. Subhojit for taking time out of his busy schedule and accepting the invitation to deliver a talk on the groundbreaking research of the Nobel laureate, Dr. Svante Pääbo. Thank you, Dr. Sen, for describing in a very lucid manner and in an engaging narrative form the significant findings of Pääbo, which aims to answer the question of what makes us uniquely human with the help of genomics. You have placed these findings against the background of earlier significant findings, and have also given us a perspective of futuristic applications. Your words have made a profound impact on the audience, as evident from the engaging question and answer session that ensued post your delivery of the presentation. Thank you once again for your invigorating talk and valuable insights that have inspired many young research minds today. I now proceed to place on record our sincere thanks to everyone who either directly or indirectly have contributed to the happening of this event and its consequent success. I begin by thanking our Chancellor, Sri Samir Somaya, Vice Chancellor, Professor Rajasekharan Pillai, Provost, Dr. Raghunath Shivgankar, and Registrar, Dr. Nandkumar Gilke, for their unwavering support and encouragement towards organizing this event. I now mention a special word of thanks to our Vice Chancellor for ideating this event, with the vision of encouraging and nurturing a research culture in the minds of our young Somayites. A big thank you to Dr. Parvati and the CIRAC team, headed by Dr. Mayuri Argeri, the Director of CIRAC and Dean Research, for making this suggestion happen. Thank you to all the HOIs of institutes in Somaya and Ayurvihar campus for their support and assistance, and for encouraging participation of their faculty and students. We are indeed grateful to all the attendees, both faculty and students from various disciplines, for their active listening and participation. A special thank you to Dr. Parvati for comparing the event and Dr. Seema Sambrani for introducing the guest speaker. An event as enormous as this would not have been possible without the support of several logistic personnel. We therefore extend our gratitude to Sri Ashok Kunder and his team for the auditorium booking and necessary logistics, to Avas Company for the chairs, to Genal Studio for the mic and technical assistance, and to Sri Amey of our IT team for coordinating with Genal Studio to make this event stream live on YouTube. We also express our thanks to the PR team for their promotional activities and the impactful flyer design. I conclude by offering my sincere apologies. Perchance I have missed out acknowledging anyone who either directly or indirectly supported us to make this event a great success. Thank you, one and all.
And I hereby thank Dr. Jacinda George. She missed her out. Thank you all. Thank you, sir. With this, the event is done. Thank you.